I think everyone has joined, but maybe there will be extra people joining later. So I want to welcome everyone to this webinar about phototoxicity. For your information, this webinar will be recorded, and you will receive a copy later later today. Before we start, just a little bit of house rules. Your microphone will be muted. If you have questions, please ask them via the chat, and we will have time during the Q&A to answer some of your questions. We will start with the presentation of Erik Manders. Some of you have heard of him, some of you know him. Erik is our Senior Scientific Executive at Confocal NL. He's the co-founder also of Confocal NL and RCM technique at the University of Amsterdam at the time. And at the end of the presentation, when he's ready, we will have time to answer the questions you've put in our chat. After that, we will go to a live demonstration of the NL5, which will be done by Anna here. We'll give you a good demo basically about the imaging of the NL5 and to see how the lifestyle imaging can occur. So without further ado, I give the word now to Erik.
Thank you for the introduction. What I would like to do
I cannot hear you, Erik. Yes.
So now it is okay?
Yes, I can hear you.
Is it better now? Yeah? So we will talk about the fundamentals of phototoxicity and then to see how we can reduce and how we can optimize the microscope to get good imaging. So maybe it's good to tell that in 1996, I started with live cell imaging at the University of Oxford at the time. And at that time, not many people were doing live cell imaging. And I found out that always the cell that I was looking at was dying or didn't go through mitosis. And I learned from people in photodynamic therapy. That's a therapy where they treat cancer cells with light and fluorophores. That phototoxicity is the problem. And so later in Amsterdam we made a method to reduce phototoxicity, and then in 2013 we started with the RCM. When we built the RCM, one of the first things that we saw is that photobleaching is not very strong and that we get very strong, bright and sharp images. So, and later we found out that phototoxicity is really a lot less when you use the rescan confocal microscope. So we wonder why. Why is that? To answer that question, we first go to the fundamentals of fluorescence, the Jablonski diagram you see here, and I have to now make my pointer. You see how fluorophores are excited by excitation light and emit a photon when they go back to the ground state. But sometimes they go to the triplet state, and the T also stands for triple, but also for trouble. Because what happens when a fluorophore is in the triplet state, it may, under some conditions, produce ROS, reactive oxygen species. And when this happens, that is trouble. That means photo damage. So that is, in fact, the background of photo damage. So it means that if I have a cell without any light, but loaded with a fluorophore, it's okay, it's fine. When I keep the cells in the incubator for a night, they are happy. When I have unloaded cells, so cells without any fluorophore, and I shine light on it, they are happy because there is nothing that absorbs light. It's only when I have the light on, so the excitation light on, and a fluorophore, then I get photo damage. And it's a pity that this is exactly the situation that we see cells. So it means that if we see cells, if we image cells, we always produce phototoxicity. And this is always the balance that we are looking for. The art of imaging is really finding the balance between cell survival and image quality. We want to have both, but this is difficult to do. So what do we know about phototoxicity? First of all, what we just saw, it is the excited fluorophore that induces the toxicity. Another thing is that it is directly related to photobleaching. So the people who develop probes for photodynamic therapy, they test not on toxicity, but on photobleaching. If it bleaches, you are almost certain that you produce a lot of toxicity. Then there is some color dependency. It means when I use UV light, it's much more toxic because UV light is also absorbed by all kinds of molecules. Far red light and near infrared light is less toxic, but so there is some dependency on wavelength. Then phototoxicity depends on the molecular structure of fluorophores. Some fluorophores are just more toxic than others. Let's say, for example, fluorescein, FITC, is much more toxic than the GFP family. And the last thing here is also, of course, the location of the fluorophore inside the cell. If it is very close to DNA, it's much more toxic than if the fluorophore is somewhere in the cytoplasm. So now the question is, how can we reduce phototoxicity? First of all, we want to reduce the light dose. And light dose is the product of multiplication of intensity and exposure time. So if I have here the intensity and the exposure time, and I expose for a certain time with an intensity, that's the light dose, this surface here. Okay, so I can reduce either the light intensity, the laser intensity, or the exposure time. Of course, also both. Another point that is important is the dose rate, and that is the first derivative of the dose, which is, we can see here, I can, in these two situations, I have the same dose, only the way I give the dose is different. Here, for a very short time, I gave a very high intensity. Here, for a long time, I give a low intensity. And the low intensity for a long time is always much more gentle for cells than a very short exposure. Okay. Then we can add ROS scavengers. That's also what people in live cell imaging sometimes do. So we saw just this picture. If I now add to the cells another molecule that easily reacts with ROS, I take away the ROS and there will be less photo damage. Then a very important one, and many people will not realize this, no visual inspection. So if I look with a rescan confocal microscope for 2.5 hours, my total light dose will be 5 joules per square centimeter. If I do visual inspection for 15 seconds, so with my eyes, I look for 15 minutes to select, this is a nice cell. Then I immediately kill the cell with 750 joules per square centimeter. If you compare this to photodynamic therapy, so where they kill cancer cells with light, there the killing dose is between 5 and 50 joules per square centimeter. So don't look at your cells with your eyes. And then, yeah, find the right balance of imaging. And this I have to explain. This is a picture that I always showed during lectures for students, and they love this. And I hope you will love this too. I will explain this by looking at image quality. In the next slide, we see here, image quality is determined by, influenced by many different parameters. For example, cell survival. If I have a dying cell, it's not a nice image. You don't want to have photo bleaching. You want to have a high sampling rate. And you want to have a 3D sampling rate, has to be so many X and Y pixels and Z stacks. Now I make these two, these one are black because this I can tune with the settings of my microscope. So these that's why these are black. I can tune them. And if I look now at the 3D sampling and the time sampling, that means that together they make the exposure time, the total exposure time that the laser is on. And the total exposure time has an influence on cell survival and photo bleaching. The longer the time, the less the cell survival, and that's bad for the image quality. Then resolution is important. That's what we want. And we want to have a good signal to noise ratio. Well, if I now look at laser intensity has an influence on signal to noise ratio. The higher the laser, the less the noise. And also on photo bleaching and phototoxicity. And in this way, you see that almost all these different components are connected. Also, the pinhole diameter has an influence on the signal to noise ratio and the 3D sampling. But there are also some properties of the fluorophore itself. I just said some fluorophores are more toxic than others, and fluorophores have a quantum yield and extinction coefficient. So all these different parameters you have to tune. And there are only 4 knobs that you can, 4 tuning points. In the standard confocal microscope, I can tune the pinhole diameter, the laser, time sampling, and 3D sampling. And now it is the art to find the right balance between all of them. So that is, I will come back to this later. That is finding the right balance between all the settings. And then another point is use a sensitive system. So the rescan confocal microscope turns out to be quite a sensitive system. And this, the PhD student Julia De Luca, at the time at the university, she researched how this is, what are the numbers. So in her paper, she mentions that you need less light because you don't, in rescan confocal microscopy, you don't have to squeeze the pinhole to get the right resolution. You also use a more sensitive camera. So a CMOS camera nowadays is 80 or 90 or 95%. The detectors are between 20 and 40%. So there we win quite a lot. And then we also have in the rescan confocal microscope a concentration of light. Because the resolution is higher, it means that the light arrives at the camera in a smaller spot. So the light is more concentrated, so the intensity is higher. So there we win also a bit. So if you measure the total, it's between a factor of 4 and maybe 8 to win. So that's quite a lot. And since we know that phototoxicity is not a linear process, the cell can cope with a little bit of light, but if it gets too much, then it starts to be unhappy, but it can cope with a little bit of light. So this 4 to 8 times, this factor 4 to 8 helps to just keep in this acceptable range of intensities. Now, another thing is important to realize. That is that sensitivity and resolution are complete different things, but they go hand in hand. There is no resolution without good sensitivity and the other way around. I will explain it here. So here you see two images in a high resolution and a low resolution made with a confocal microscope. So this is with a small pinhole, pinhole of 0.5, and the other one with a pinhole of 2. And you see that the more noise, so the weaker the image, or the weaker, the lower the sensitivity, I see it becomes more and more hard to see these objects and to see the resolution. In the rescan confocal microscope, the sensitivity is 4 times higher, so it means the signal-to-noise ratio is much better, it's less noisy, and that means that we can also see the resolution. So that helps also. It also helps to see the crisp and sharp images. So it means that when we go back to this balance of imaging slide, that in rescan confocal microscopy, the signal-to-noise ratio is higher. So it means that, according to this line, I can go lower with a laser intensity to compensate, to accept. I accept a little bit more noise, so I reduce the laser. And then that means that I get less photo bleaching and a better cell survival. So that's why sensitivity, signal-to-noise ratio, and cell survival are connected. So it means it is possible to get a good cell survival and a good signal-to-noise ratio, imaging quality. So to show that, we see here some cells stained with a mitochondria staining, and if it will start. Wait. I think I have to use the pointer. Yeah. So it starts. You see that the cells easily go through mitosis, but you also see that when the cells are in mitosis, the intensity is a little bit lower of the whole image. Here you see the intensity. The intensity stays more or less the same. And this is for hours and hours. The whole image is, the series is 61 hours, and the laser power is only 85 nanowatts. So it's very hard to see the spot of light on your coming out of your objective. So this it's clear the photo bleaching is almost not there. And then the other point, what we can see in the same series of images, you see here the mitochondria, and people in the audience who are experts in mitochondria imaging, they will recognize, here you see the deconvolved image, this is the not deconvolved image, that the shape of these mitochondria are not changing, and you recognize these cells are completely happy. So it's the same movie, but another part of the movie we took to look at the mitochondria. Okay, so I come to the conclusion: the rescan confocal microscope has 4 to 8 times higher sensitivity compared to standard confocal microscopes, and this high sensitivity means that we can use a lower laser power, and that means a lower phototoxicity and lower photo bleaching. And that, in the final conclusion, you can say that RCM helps really a lot to be gentle for yourselves. Now, if I have some more minutes, is that okay? I want to show a recent paper that came out at the University of Amsterdam, the microbiology group of Stanley Brull, and they looked at the germinosome, which is a little group of proteins and molecules that are kind of the nose of the bacterial spore. So when bacteria are in bad conditions, they form spores, and there are now molecules on the surface of the spores that sense, there is still no food, and after a while, maybe, yes, there is food, now we can become a bacteria again. So it was always thought that these molecules are in groups at the surface of the of the spore. So this is a spore which has the size is something like 1 micron. So it's a noisy, or it's a blurry image, but you have to realize from left to right is more or less than, is about 1 micron. But now we looked at the same spores, living spores, dormant but alive, with a rescan microscope, and we tuned on a very small area, only on one spore, so we could increase the frame rate to 45 frames per second. And we made a short image of 20, something like 20, 30 seconds. And what you see now is that these molecules are floating around in the on the spore, and sometimes you see, if you look well, you see sometimes a very sharp intensity, very high intensity, short spot, high intensity. So it means that the molecules travel along the spore membranes and are making a clump sometimes, but so it means that we get a complete different view on how the germinosome works, what it looks like. It's not a static thing like this. It looks like this with a wide field microscope because we average over all these images for the duration of 2 seconds. So this gave, for this paper, a complete new view on how germinosomes are formed. So I didn't want to, I want to show you this because this is quite impressive. So the extra conclusion here is high speed, high sensitivity, low noise, low toxicity imaging reveals the dynamics of at a molecular level. So that is, I think, what I want to show this time. Yeah. Oh.
Yeah. Thank you very much, Erik. There's a little bit of a discussion on the chat.
Oh yeah.
So I think there was trying to summarize it. There's a lot, but for some of the questions they had. It's about the phototoxicity caused by the ROS and what is and regarding the excitation. I think what I understand from here is that what they are looking at is what proportion is caused by ROS generated on which part is by the light dose, but I didn't think we have an answer on that. That's one of the questions there.
The light dose compared to ROS. Yeah, it is very hard to say how much ROS exactly is produced. It really depends on the fluorophore itself. What I just said, for example, fluorescein is much more toxic than GFPs. When I did this work on live cell imaging in '96, it was almost impossible to get cells through mitosis. Later, I worked with GFP. It was much easier. And also the people who developed probes for photodynamic therapy, they, for example, put a lot of halogens in the molecule to make it more toxic. So exactly how much, what is the relationship, is hard to say.
Okay, thank you, Erik. I hope that answers your question. If not, we're always afterwards. We can have an email or a discussion about that. I had a question about, you were mentioning the visual imaging. You were saying that it's very phototoxic. Could you elaborate a little bit on that?
Well, it's it's the visual imaging which takes too much time. Our eyes are quite sensitive, so we can see very dark, low light levels. But our eyes, it's not our eyes are bad, our memory is bad. So we always, to see something, we need more and more, continuously, more photons in our detector. A camera only needs for a split of a second photons and then puts it in the memory and places it on my screen. So um, with with camera illumination, I can be much more gentle for cells than looking with my eyes. So really, never look with your eyes to cells that you want to image. It's really shooting what you do. Um,
And I guess now with the fast confocal NL5, that makes it also easier.
Yeah, so the faster you can image and the shorter the exposure time, the better it is. So with a point scanner like the RCM2, it's good, and also the NL5, it's even much more faster. I think that helps a lot.
Okay, again, thanks a lot, Erik. So that's a nice bridge to the next part. We will go now to the part that we will live demo the NL5. So Anna, please, if you can take over. The floor is yours.
Thank you very much, Erdal. So I will just quickly share my screen. Yes, so as Erik already explained, RCM and NL5 are ideal for preventing phototoxicity if you are doing microscopy imaging. You can see an RCM2 in my background, but for today's demo, I am going to use NL5. And first of all, what is NL5? So in the background, you can see a setup with NL5. NL5 itself is an easy-to-use confocal unit, couples to any inverted or upright microscope, and turns it into a highly sensitive, fast scanning confocal system, which is ideal, for example, for deep 3D imaging. For NL5, we are using a highly sensitive camera. That's how we can achieve using very low laser power to prevent photo bleaching and toxicity. So for the demo, for acquisition, I am going to use Micromanager, which is an open source software which controls NL5. And also it allows you to control any microscope, camera, and laser that you want to use for your experiment. So as I suppose you maybe cannot see this panel, but I'm going to use mainly the controls of NL5. So in Micromanager, I can adjust the laser power of what we are using for imaging. And I can also adjust the scanning speed. NL5, as I mentioned, is a fast scanning confocal system, which offers you to image with 25 frames per second. That's what we will use today. So I will show you how fast it is. And also, I think a very useful tool that we can switch from NL5, from confocal to epi, and also back. So if you need to find your samples and it's easier for you with epi fluorescent microscope, then we can just easily switch to the epi mode. So for that, I'm going to show you first how the bypass, so the epi fluorescent, works. So if I click on live, I do live streaming now. You can see the lookup table and also the image of the sample. Today, I am using a Volvox globator, which is a green algae. And in the sample, you can see the single cells and also the reproductive daughter colonias. So now I am going through the sample. This is a very, very thick sample. It's more than 200 microns. And of course, because this is an epifluorescence image, this is quite blurry and not. And also you can see the autofocus light and the low contrast. But we can do, for example, with a micromanager, we can do a multidimensional acquisition. So we can just quickly scan through the whole sample. For that, I am going to go down the sample and then find the bottom and the top position for doing Z-stack. Yeah, and I will choose, for example, 1 micron step size. And we have, in the end, we have 54 slices. So if I click on acquire and change the contrasts with the lookup table, then you can see where we are doing the imaging with EPI. So let's go to NL5 because that's we want to see today. It's just an easy click and then we can already go live. Of course, it's better to choose the same color for that. Here it is. So now you can see a confocal image with NL5, which has way higher resolution. So NL5 offers the same resolution as a standard confocal microscope, which is 240 nanometers on a raw image and 170 nanometers after deconvolution. So let's repeat the same experiment with NL5 with the same settings. If I click on acquire, you can see how fast it scans even in the Z, because of course, when you are going deeper in the sample, you are always a bit slower. But how fast this is? So we had 54 slices, stacks, and it was 2 seconds to scan through the whole sample. And now I will compare it to the bypass image. So for that, I would like to choose the same plane, which is 15. And you can see the difference. So NL5 offers a way higher resolution and high contrast image. And also, if we are going deeper in the sample, then we still have, so even after 19 microns, we have a very high contrast and high signal to noise ratio image with NL5. So since it is, I just need to find the right window, exactly. So since it is a fast scanning confocal microscope, it scans and it scans very fast, not only in the Z, but also in X and Y. So now I am just finding the right place, what I would like to see, and you can see how fast it is. You can just quickly scan through the whole image in X and Y and Z and to see what you want to see. And also it's ideal for tiling and stitching because of the high uniformity. So if you see the intensity, the light intensity of, for example, here in the middle, if I bring this daughter colony to the corners, even in the corners, you get the same intensity. So you get a nice image while you are doing stitching. So here you could see how fast NL5 is, but what can we use it for? And I will go back to Webinar Jam and then show some nice videos. So for example, it can be used for long-term time-lapse imaging. This is a zebrafish embryo, which was imaged for more than 24 hours. You can see the endothelial cells, how they are moving. So this is ideal for developmental biological research. You have a large field of view, you have high resolution, and you can just scan through the whole sample if you want. But NL5 can be also used for high content screening. So if you have if you want to do multi-channel imaging, you have multiple colors, then you can just quickly switch from one color to the other. So this is tubulin of HUVEC cell, the actin, and the DNA. And NL5 can also be used for the studying fast cell dynamics. So this is a HeLa cell, which is stained for actin. And you can see how the cell is sensing the environment where to go. Thank you very much.
Yes, thank you, Anna. Thank you for the demonstration. It was really nice to see that in a live setting you were able to go in X, Y, and in the Z, and it really looked like there was no lag at all. So it was really nice. Thanks a lot for that. I did not see any further questions, so it was very clear everything. So I hope you all enjoyed it. If there are questions which were not answered, you can always get in contact with us. I'll just open it and start the presentation. Yeah, correct. Great. So thank you everyone for joining us. And yeah, as I said, please feel free to contact us for demonstration online or on your site. I would also check the offer area of the webinar, where you can see some of our products, which will help you get your phototoxicity down. You can look it up in our web shop also. So then going to the next sheet. I'm trying to get used to this one. Okay. We will have a next webinar about organoids in collaboration with Dr. Daniel Kruger of the Hubrecht Institute from Hans Klevers Lab. We will do that in March. The exact details will follow. So please connect with us on LinkedIn so you're up to date of the newest webinars. And we'll try to inform you as soon as possible. So again, thank you much for joining us, and looking forward to seeing you next time. Bye-bye.
Thank you. Bye.